If I say something that you don't understand, or if some things are difficult to follow, just raise your hand, and if you have any questions, you can also ask me. So, the main thing I want to show in this lecture is how the effects of oxidative stress can be used in an epidemiological context. And I will do this by explaining my research, and this is about how environmental factors, including air pollution, induce oxidative stress during the pregnancy of the mother, and how this can be translated to clinical outcomes that can arise later in life. Also, I just want to mention I haven't put my slides on Blackboard. I will put them later on Blackboard, because I think for the exam you can just scroll by my PowerPoint and then you have all the information you need. No, but it's just, it isn't a lot of theory, this lecture. It's just how is research performed, how can we use the things that we have learned in theoretical lectures. Okay? I will first give some an introduction about epidemiology, biomarkers, and air pollution. So I just want to begin with the boring part. That epidemiology, it's a word that consists of three parts: epi, upon, demos, people, logos, study. It's just a study upon people. And what epidemiology wants to search is how diseases are distributed upon the general population. A few examples of epidemiology are the association between asbestos exposure and mesothelioma. Mesothelioma is a cancer of the internal lining of the internal organs, such as the pleura of the lung. That's the spot where mesothelioma occurs most frequently. Also, the exposure to softenon or thalidomide, I think everybody knows it. It gives birth defects. So thalidomide was given to pregnant women that were nauseous during pregnancy, and later on they saw that exposure to thalidomide gives birth defects. There are some really simple examples of the importance of epidemiology. And one more is the exposure to smoking. I don't know if anybody smokes here. No? No, but the association between smoking and lung cancer is another example of epidemiological research. Now, traditionally, epidemiology sought to link the external exposure to a disease, but a few years ago, or decades ago, they had not the ability to investigate the intermediate processes. The intermediate processes, I mean, what is happening inside the body, what effects occur if somebody is exposed to a chemical or smoke or so on. And the molecular epidemiology arose, I think, in the 70s, where one gained more experience in the intermediate processes, also technology, gene expression, proteomics, and so on, where they elucidated the intermediate processes from exposure to the clinical outcome. Some structural changes that occur in organs or on the molecular level, and so on. They make use of three different categories of biomarkers, biological changes that occur, and these are biomarkers of exposure, biomarkers of effect or disease, and biomarkers of susceptibility. Now I will go on into more detail about these biomarkers. So it's just an approach to incorporate molecular, cellular, and other biological measurements into epidemiological research. The first category are biomarkers of exposure. These are used to just measure to which extent one is exposed to a chemical or a metabolite or so on. An example is the urinary gotinin. Gotinin is a metabolite of nicotine, and if you smoke, you can measure the gotinin level in urine, and then you can see does anyone have smoked throughout pregnancy or during life or so on. That's the most important example, and if you remember this, then you know enough about biomarkers of exposure. Now, these can be measured in different matrices. I don't know if I state right, matrices, yes. These are, for example, hair. You can use fingernails, toenails. The most prominent used matrix is urine, but also blood. The disadvantages of blood is that it is invasive, so you have to put a needle inside somebody's arm and pull the blood out, because with urine it isn't the case. Also, another example of urine is the recent news about the EPO cycling affair. They measure metabolites of EPO in the urine, and that's also an example. In my research, I use placenta and cord blood. The main advantage of placenta and cord blood is that we look to the exposure of the unborn child, so that's the only reason why we use this, and it's the most representative for the unborn child. The disadvantages is that it can only be obtained by only a short period during their pregnancy and not in another period. Okay, biomarker susceptibility. I don't have to say much about it because it's just the natural characteristics of a person. So if you have a mutation in a biotransformation gene, for example the GSDM1 gene, or for example the cytochrome P450 genes, there are genes that are responsible for biotransformate certain, for example, benzene levels, so that they can be excreted into the urine. So if you have a polymorphism inside a gene, it does not work properly, and you have more risk for example, lung cancer. Then the biomarkers of effect or disease. These are quantifiable changes, for example, in DNA damage. And biomarkers of effect can be observed at different levels. The first level is at DNA level. You can also measure at RNA level. It's gene expression. You can also measure at protein level, the proteomics. And last, at the metabolite level, so metabolomics. And I want to give an example from this one. It's a really useful example and a really interesting example, because the current methods to look at breast cancer for women is at the age of, I think, 50, 60, so approximately. And they do this by taking a mammography of the breast, and then they see if there are little spots in the breast that may be linked to breast cancer. Now, here in Hasselt, they have developed a method by which they take blood and they search for a certain type of subset of metabolites that can be linked to the development of breast cancer. And the main advantage of this technique is that you can detect breast cancer in a very early stage of 30 to 40-year-olds. So that's quite an amazing discovery. And in my research, I focus more on the DNA and the RNA level. And I measure telomeres, mitochondrial DNA, DNA methylation, and gene expression. But I will come back to that later. Okay, so I have told you something about oxidative stress, the biomarkers that we are going to measure. And now I want to tell you something about air pollution. Yeah, these slides are not that important. I wanted to tell you something about it, but in these slides it just says that Air pollution is a problem that arose a few decades ago with the upcoming of the Industrial Revolution, where many factories have been built and the air pollution levels rise extensively. And here you can see at a date, it's a graph of 1952, so the exposure levels increased and also the immediate deaths increased. But this is not a problem of the past, this is also a problem of the present. As you can see here in these research articles, elevated DNA damage with youngsters in Genk in September 2011. October 2000, yeah, there are articles that I have used last year, so I haven't got some recent articles. Belgian air is the most unhealthy of them all. Yeah, it's really true. But don't worry, our Dutch, our Netherlands friends are also doing very worse. They have also high exposures to air pollution levels. But not only Belgium and the Netherlands, but also the entire Europe has too high air pollution levels. This is a map of 2002, but I have looked through the maps of 2006 and 2010, but they look quite the same. So this table depicts the guidelines of air pollution, and this is the World Health Organization and the European Union. So you can see there's a different number of guidelines to which one makes the legislation. And this difference arises because the World Health Organization bases on research that's performed on the entire globe, whereas the European Union only looks at research performed in our Europe region. So it's important to notice this difference. Okay, what is air pollution? Air pollution is a mixture of gaseous pollutants, but also particulate matter. Particulate matter or fine dust, fijn stof, zoals we zeggen. It's a heterogenic substance with particles that vary in aerodynamic diameters. And I just want you to visualize particulate matter. You need to visualize it as a sort of sponge that contains all sorts of toxic metals, but also particles from natural sources, but also from anthropogenic sources like the combustion process of diesel exhaust and so on. It's just like a sponge that contains toxic particles. And we can divide it in certain subclasses. We have the PM10, that are all particles that are smaller than 10 micrometers. I made here a figure where it's represented to human hair. I hope you like it. But the crude particles, the PM10, I hope you can see. No? Okay. These contain more the natural sources, but also the anthropogenic sources. So it's both contains like sea salt, pollen, windblown dust, but also this category, if you understand what I mean. Then the PM2.5, that are all particles that are smaller than 2.5 micrometers. And these contain more the particles that are retrieved from the combustion processes, from cars, from factories. You have the nitrates, sulfates, also toxic metals, and so on. And then you have the ultrafine particles, and these particles are really the very small particles from the combustion processes, and diesel goed. So this is what particulate matter is. Particulate matter has been shown to induce reactive oxygen species and inflammation. Now, the process of the formation of reactive oxygen species, I'm not going into detail, but you know that it is with the formation of superoxides and hydrogen peroxide and so on. Yeah, you know all about it, okay? And it's been able to induce DNA, lipid, and protein damage. Now, the effects or the health effects of particulate matter are contributed to two things. You have the chemical composition, as I've already told you, but you have also the particle size, and the smaller the particles are, the deeper they will penetrate into the lung. Here is a picture of a lung with the alveolus at the end. And in this graph you can also see it. So the larger particles are stopped by the defense mechanisms in the throat. And they are cleared by a mucociliary defense mechanism, actually. And the small particles, they can reach the alveoli. And the really small particles, they are even able to diffuse from the bloodstream, from the lung to the bloodstream, and exert their hazardous effects like reactive oxygen species and inflammation. And this is also the reason why it's interesting to look at placenta tissue, because a mother inhales these particles, it traverse or they translocate to the bloodstream, and then maybe they can affect the placenta tissue and the umbilical cord blood. Okay, and another thing is that small particles are, they stay longer in the air, so they are airborne longer. And this is a computer simulation of the concentration of PM2.5 and the visibility. So the longer or the smaller the particles are, the longer they stay in the air. And you can also see, observe this effect also in very busy cities like Shanghai, for example, or in New York. You can see that the visibility is really less than when there's less air pollution. Okay, this brings me to my hypothesis from my PhD project. So exposure to environmental factors and lifestyle factors, also smoking, diet, exercise, fish intake, and so on, they can affect molecular markers of aging, telomeres, mitochondrial DNA, that alter the in utero development and lead to adverse pregnancy outcomes, such as low birth weight. And ultimately, this is a risk factor for developing diseases that arise in adult life, like arterial stiffness and asthma. And we want to do this because we want to prevent events like heart failure. So people that have a low birth weight, for example, they have a lot more risk to develop cardiovascular diseases in later life. I hope this is clear what the main project line is. Okay. Then we go into a little boring part. I hope I still amuse you with my lecture. No, but it's just to show you that research is not only the molecular thing and in working in the lab and so on, but also some clinical parts, some sample collection and so on. So we first need to set up a birth cohort where we collect the placenta tissue, cord blood, and maternal matrices, such as blood and urine. Then we needed to measure the exposures to which pregnant women are exposed during pregnancy, like fine dust, toxic metals, and smoking. And then we measured some biomarkers like the mitochondrial DNA content, DNA methylation, and telomere length. And finally, we want to do the health evaluation, as I have already told you here, a health evaluation of these young children to see if they are more prone to cardiovascular disease. Okay, then the methods. So we have a collaboration with the hospital East Limburg in Genk, where we retrieve every weekend the placenta tissue, umbilical cord. So these are the matrices that are. Do you have a question? I thought you would raise your hand. Okay. They are representative for the baby. We also collect maternal blood and maternal urine. That's really a picture of a placenta. I don't know if somebody saw a real placenta in real life. No? That's how it looks. Okay, why do we use placenta tissue? You can see here a photo of the placenta with the umbilical cord that connects the fetus with the placenta. You have here the the fetal side and the maternal side. And this maternal side is attached to the womb of the mother. So we use the placenta tissue because it's very representative for the fetus. It's also the main role of the placenta is the development. So if the placenta does not work properly, the development of the fetus may also be influenced. And it's also a non-invasive tissue, or we can collect it in a non-invasive way. We obtain RNA and DNA from the placenta, RNA for gene expression, DNA for these other three things. We use urine. I have already told you how you can measure metabolites. It's also non-invasive. And then we collect cord blood and maternal blood. The reason why we collect both of these blood types is that we want to see if there is a difference between the exposure of the mother and the exposure of the fetus, because I already told you the placenta forms the barrier between the mother and the fetus, and if we can see that exposures traverse the placenta, it should also be how do you say it? It should also be present in the umbilical cord blood. In blood, you can also measure all types of contaminants, such as gases, solvents, metals, and fat-soluble compounds. That's the main advantage of using blood. And you can also extract DNA, RNA, and so on. Okay, I just have half an hour, I think. The next thing we needed to do is measure some exposures. It is not possible to measure the exposure to air pollution from each woman that is pregnant for 9 months. It just is not possible, and therefore we use a pre-interpolation method. And this method is based on that we collect data from, I think it's 61 measuring stations that measure particulate matter in all around in Belgium. And then they, with sophisticated models, they extrapolate these values to, for example, a mother that is living here in Diepenbeek. So they extrapolate these values to another spot, and they take into account the air temperature, but also the wind and so on. So it's a really validated model. If you don't believe me, you can look up the article where they provide the evidence that it is a validated model to use. So this makes us able to ask all the exposure levels of all mothers during the 9 months that are living in different spots. And these exposure levels, they are provided in 5 by 5 kilometer grids, but they are improving in 1 by 1 kilometer grids. So that's, you can say, okay, that's quite crude, so roughly, but that's true. But the exposure levels don't vary that much. If I stand here and here, the exposure level is the same. It's just like with this model. Okay, then we also measured this distance to major roads, and I think the Belgian students will know the program also tested on humans. They did a little experiment by putting on a woman some device that measured the ultrafine particles to which she is exposed, and then they told her to run. First, she ran across the countryside. So you can see here, these are the background levels. These are very low. So if she ran in the city, you can see here that the exposure levels peaked here in a very busy cross section in Antwerp. This was. So this is just to show that the proximity to a major road is really important to measure. Okay. I will just read that. We also measure some lifestyle factors, and we do this via the questionnaire. We ask these mothers, did you smoke throughout pregnancy? What was your diet? Did you exercise a lot? And so on. We also measure toxic metals in the blood via ICP-MS. That's really important. Okay, now the results section. In my PhD, I started it with almost nothing, and I made the cohort grew by approximately 400 newborns, and I already measured the mitochondrial DNA content and DNA methylation, by which I will go into more detail now. So these are the first results of my PhD project, where I'm very proud of. I hope you're equally enthusiastic as me. Okay, the research aim 1 was if in utero exposure to particulate air pollution, PM10, led to changes in the mitochondrial DNA content of placental tissue or cord blood. So what is mitochondrial DNA content? You all know mitochondria that are intracellular organelles that are that provides all cells of the necessary energy through the production of ATP via the oxidative phosphorylation. Now each cell contains 200 to 2000 of these mitochondria, and each mitochondria contains 2 to 10 copies of mitochondrial DNA. So this is circular DNA that exists independently of nuclear DNA, and that's very important to keep in mind. So the mitochondrial DNA content is a molecular marker of mitochondrial damage and mitochondrial inflammation. And why this is, I will explain in this figure. So here we have the mitochondria of the mitochondrion with the mitochondrial DNA. And reactive oxygen species are formed as a byproduct of natural aerobic respiration, but also by environmental components such as particulate matter, toxic metals. They induce reactive oxygen species that can translocate into the mitochondria and induce mutations to the mitochondrial DNA. Now, the mitochondrial DNA is very susceptible to mutations, and this is very important. You should remember this because maybe it's an exam question. The mitochondrial DNA lacks histones. It has also an insufficient DNA repair and also lacks introns, and that's the main reason why mitochondrial DNA is very susceptible to mutation, and it can be used as a biomarker. Okay. Now, if we have these mutations, we can get an energy compensation of the mitochondria. We get an increase in mitochondrial abundance, also an increase in the mitochondrial DNA content. But because this is full mutations, we can get a reactive oxygen species feedback mechanism, which ultimately leads to a decline in the mitochondrial DNA and also the mitochondrial abundance. Now, both a decrease and an increase in the mitochondrial DNA content has been associated with mitochondrial dysregulation. And the purpose of this research was to investigate if we have mitochondrial dysregulation. Is this observed in, for example, the umbilical cord rod or placental tissue, and how can this be translated to the fetus? Okay, and I have published an article about it, which I'm very proud of, in environmental health perspectives. This is one of the leading epidemiological magazines. So we investigated placental mitochondrial DNA content and particulate air pollution during in utero life. I also want to mention this is work that I performed during my last year, during my thesis year. So you're now in the 4th year, I think. Yes. So that's just a thing. If you work, if you have ambition, if you work hard, you can achieve a lot. And this is the thesis is I think from November until June. So in a few months you can do really a lot. That's just what I wanted to say. This research was performed on 178 newborns. Partition rate was 47%. Now what I did was I collected umbilical cord blood, placenta tissue. I extracted DNA, did a real-time PCR. I'm also not going into detail about the technique of real-time PCR because you all know it. But with the technique of real-time PCR, we could measure the mitochondrial DNA content. And this is measured or calculated by the ratio of the mitochondrial DNA compared to the nuclear DNA. So that's why it's very important to know that the mitochondrial DNA exists independently of nuclear DNA. The main problem was the primer selection, because mitochondrial DNA is double-stranded circular DNA. We needed to develop primers that are very specific for mitochondrial DNA. And that's what we did here. It's a table from the article. We developed 2 mitochondrial DNA primers and 3 nuclear DNA primers, and with the means of these results we could calculate the mitochondrial DNA content. This is a table from the study population characteristics. I just wanted to show you this because this number intrigues me a lot. This is mothers who continued to smoke during the pregnancy. 28 mothers of the 178 still smoke during pregnancy. I think it's certainly in this time, it's yeah. I don't agree with those mothers. So it's 15% of the total population that says that they have smoked during pregnancy. First, we performed a linear regression analysis, just a correlation plot. On the x-axis, we have here depicted the PM10 values, and on the y-axis, we have the mitochondrial DNA content in placental tissue. This is the PM10 exposure during trimester 3, the PM10 exposure during the last month, and PM10 exposure during the last week. And in these plots, we see that the higher a mother was exposed during pregnancy, the lower the mitochondrial DNA content was. So you need to recalculate that early slide about if you get an increase or a decrease in mitochondrial DNA content, this can be associated with mitochondrial dysregulation. So what we saw here was that exposure during particulate matter during the last period of pregnancy is associated with a decrease in the placental mitochondrial DNA content. If we perform the multiple regression analysis, the difference between a linear regression analysis and a multiple regression analysis is that we adjust for several factors that could influence the placental mitochondrial DNA. So if we adjust for all these factors, we still see that for each 10 microgram per cubic meter change in PM10 exposure during the last month, we have a decrease of about 20%. This was also seen in the last week. So this is placental tissue, and this is always the umbilical cord plot. This was also seen in the last week, and was also just significant in the trimester 3. These lags are all days before pregnancy, so it's this is the day of delivery, this is 1 day before delivery, this is 2 days before delivery, 3, 4, 5, 6, 7, and this is the mean of the lag, so the last week before delivery. You can also see that we did not observe an association between umbilical cord loss. Here's a table. It tells you quite the same as this figure, but I just want to point out here that we also measured the distance from residence to a major road. And important in this is that mothers that were living farther away from a major road, like an N or an E road, they were exposed to less particulate matter, and they had higher mitochondrial DNA content in placental tissue. And this confirms our hypothesis of also the used model. So distance to major road is a surrogate for traffic-related air pollution. Okay. The second thing that I investigated is DNA methylation. So the research aimed to, don't worry, I will not going to tell you much about it, so I will be done in, I think, 10 minutes. The research aimed to was that in utero exposure to particulate air pollution, in this case we measured PM2.5, lead to epigenetic alteration in placental tissue. And we only investigated placental tissue because in the previous research aim we only saw effects in placental tissue. Now, I don't know if you got a lecture about this, epigenetics. I can go on about it. You just need to know DNA, at DNA level, DNA methylation is important, at protein level, histone modification is important. So what I measured was DNA methylation. Cytosines are methylated to 5-methyl cytosines. Yeah, you already saw that, so you know it. But important in this is that hypermethylation at the promoter level, so hypermethylation of these CpG islands that are normally not methylated, can give gene silencing. And an important example is, for example, the tumor suppressor gene, T53, I think. This can be silenced by external exposures. For example, if you are exposed to benzene, benzene is able to change methylation patterns. If this is hypermethylated, the tumor suppressor gene is silenced, and this will increase your risk for developing cancer. You have also hypomethylation at the global DNA methylation, hypomethylation at global DNA level, and this is associated with chromosomal and genomic instability and loss of imprinting. And you should remember that hypomethylation at global level is chromosomal instability. Now, because I'm interested in the fetal development, it is important to know how DNA methylation patterns are formed. So you have a demethylation in the germ cell production, and also, again, a remethylation. So if the sperm cell reaches the egg cell, you get a fertilization, and then you also have a second phase of demethylation, where a remethylation starts by, at the, I think it's the morula stage, you get a silent remethylation that ends with the implementation stage. So throughout the whole life, these methylation patterns are stably maintained throughout life of an organism. So they can also be influenced by some environmental exposure, but generally they are maintained stably throughout life. Okay, I've written also a paper about this, placental DNA hypomethylation in association with particulate air pollution in early life. This was performed on 240 newborns. We did an LC-MS analysis to determine the global DNA methylation level, and this was calculated as a percentage. So we did not look at promoter level, but at global level, and as you already seen, global level is can be associated with genomic instability. We did also a linear regression analysis. We have here the exposure levels, PM2.5, and on the y-axis we have the global methylation level in percent. And here you can see that exposure to high levels of PM is associated with a lower global DNA methylation level. And if we specified our research or our exposure to trimester 1, we saw that there was also a decrease. And in this beautiful 3D plot, you can also see a major decrease during the early life of, or during the early life of the fetus in placenta tissue. Okay, we also did a multiple regression analysis. Here the results are again repeated. You have in trimester 1 a decrease, a relative difference of minus 2.47%. And this analysis was done just specifically to investigate this pattern of remethylation during the development. And we saw that the DNA methylation was affected during the implantation window, but also in the post-implantation window. And as by wonder, this is affected here because this is just the time when the placenta starts to form. So global DNA methylation level is affected during critical stages of implantation, the moment the placenta starts to form. This. The main message of this article was that in early pregnancy, DNA methylations can be altered due to the exposure of particulate matter. So future studies should elucidate the genome-wide, yeah, my English is not that good, but okay. They should elucidate genome-wide and gene-specific methylation patterns in placental tissue. And these gene-specific methylation patterns, they relate to the hypermethylation that is associated with gene silencing. Okay, the key findings are placental mitochondrial DNA content is associated with exposure to PM10 during the last period of pregnancy, and also exposure to PM2.5 lowers the degree of placental methylation. Now, whether a lower mitochondrial DNA content and altered placental methylation predicts the risk for cardiovascular disease or asthma and later in life must be further elucidated. And that's what we are planning to do in the future. So we have our birth cohort, so we are with 400 newborns and we are still continuing to grow. We have the exposure measurements, we have biomarker measurements, we have already measured the mitochondrial DNA content, DNA methylation. We will also measure the telomere length and do a health evaluation of these young children. So they will come to a follow-up visit where we will measure the arterial stiffness and see if they have developed asthma or wheezing and so on. Some future perspectives, telomeres, we have already seen this. Do everybody know? Does everybody know what telomeres are? Yes? I don't see hands. I think everybody wants to go home. No? Do we have any classes in the afternoon? No. Okay. Then I will get going. Yeah, it's just some basic characteristics of telomeres. But it's interesting to look at telomeres in the placenta tissue because with each cell division, telomeres shorten. And if we can see if telomeres in the placenta tissue or cord blocks are shorter in pregnant women who are exposed to more particulate matter, then it also confirms our aging hypothesis. Okay, some future perspectives. I already told you we will invite these little children for a follow-up study where we measure the intima media thickness. So you have here the endothelial lining, the smooth muscle cells, And the stiffer the arteries are, the thicker how do you say it? The intima media thickness will get. So it's just a measure of how susceptible somebody is for the development of cardiovascular diseases. And this is done by an echo toestel where this is my artery from the Holdes. I have very good thickness because I'm still young. With old people, you see that the intermediate thickness is very enlarged. So this is what we want to do with very young children. I'm just going to conclude. The takeaway message is you don't have to wear these masks because it is not possible to avoid air pollution. You are just exposed to it, and that's the thing. What you can do is improve environmental legislation. We can protect the unborn child. Everybody gets a fresh start. We can use predictive markers, prevent also chronic aging diseases, and lower the cost in healthcare. I just want to emphasize the importance of epidemiological research. And these are the points that you have to know for the exam. It's just that you have a global picture of how biomarkers are used in an epidemiological context. Last but not least, I want to make a call for junior projects. You must, I think you have to choose in October, November, I think. November, yes. You have to choose for a junior project, and I would certainly recommend my project. No, because as I said, it's very interesting to just combine the experimental laboratory work, but also the clinical part. So we will measure, we will use gene expression, real-time PCR, but on the other hand, we will investigate these young children. We will do an actual. We will ask these mothers to come by. So it's really interesting to learn something besides the laboratory. And I also want to emphasize that it is not possible to choose this subject because I know I will get a lot of applications, I think, no? It's not possible to choose this for a junior project and year after you will need to choose a thesis project, and it's not, I don't know if it's the same in Maastricht, but in Hasselt it's not possible to get a project 2 years at a row. So you just need to keep in mind. Then one last thing, if anybody has some questions? No? If somebody is interested in my project or maybe another project, because this is not the only project, I just want to make some commercial. We have also a project that is dealing with exposure to environmental factors and cognitive function, so the neurobehavioral part. We also have a study on elderly that also investigates environmental factors on more older population. So these projects will also be available on the Blackboard system, I think, and you can just choose. If anybody has a question or wants to have more information about this project, you can always send me an email. I am on the Hasselt site, so you can always contact me. Thank you for your attention.